Hello everyone, in this lecture we'll talk about pyruvate and the citric acid cycle. So if you remember, the end product of glycolysis is the 3-carbon pyruvate. So this 3-carbon sugar molecule will have another extra step to be converted into the 2-carbon acetyl coenzyme A that will eventually fit into the citric acid cycle for further oxidation. For information, the citric acid cycle is also known as the Krebs cycle or the TCA cycle, which stands for tricarboxylic acid cycle. Okay, here is a brain teaser from our last lecture. What would be the consequences if a 6-carbon sugar was converted into a 4-carbon and a 2-carbon sugar instead of 2 3-carbon sugar in glycolysis? Well, you will need two sets of pathways and consequently two sets of enzymes to metabolize the 4-carbon sugar and the 2-carbon sugar, which means you will have to invest more energy to produce two sets of enzymes, and this makes the whole process less efficient. So in this lecture, there are 4 learning objectives. Number 1 will describe the control strategies of glycolysis by modifying the activity of the enzymes. And then we'll look at different fates of pyruvate depending on the needs of the cell. And number 3, we'll try to appreciate the role of vitamins in metabolism. And last but not least, we will outline the important steps in citric acid cycle. We can say that all forms of power generation requires control of output for at least 2 reasons. Number 1, we would like to maximize efficiency. So that is to match supply according to demand, and therefore no power will be wasted, and that is to reduce pollution as well. And the second reason is that we would like to maintain a stable environment because power generation is a very complicated process, and if anything goes haywire, you will see the whole system being affected. Likewise, our metabolic pathways such as glycolysis must also be tightly regulated. The first reason to do that is to make sure that the supply of ATP is linked with demand. For example, your body will produce more ATP when you're having a very hectic physical exercise. And for certain mammals that undergo hibernation, the rate of metabolism could be reduced by half to conserve energy. While a second reason we need the control of glycolysis is to allow cells or organisms to respond to environmental changes, such as changes in temperature, diet, and microenvironment. So it's all about adaptation. For example, the rate of energy production must be adjusted to the environment, such as in the winter, you're going to need more energy than in the summer, and so on. While a third factor is about maintaining a constant internal environment in a process known as homeostasis. So you can see human body as a tightly regulated system. So parameters such as body temperature, blood pressure, or even our chemical constituents are maintained within a small range of values. That's because for enzyme to function optimally, these physical conditions must be controlled. And therefore, glycolysis must also be regulated to make sure that the internal environment, especially our body temperature, is well regulated. Well, last but not least, in glycolysis, we need to have interaction in between different tissues, such as the liver, the adipose tissue, and muscle, because these tissues, they are involved in different parts of glucose metabolism. As I mentioned, glycolysis is catalyzed by a set of enzymes. So by adjusting the rate of enzymes, we could achieve the control of glycolysis. And usually, it is the enzymes at the start of pathway that are first regulated. Because by doing so, all other pathways and enzymes in the downstream are regulated at the same time. While a second strategy is to control enzymes at specific branch point. Because very often, we have branches in biochemical pathway for the synthesis of specific substrates. So if we do not need to switch off the whole pathway, then we can switch off that specific process by controlling the enzymes at the branch point. And to adjust the rate of reaction or to adjust the activity of enzymes, we have two general strategies. First one, we can control the amount of an enzyme. Logically, when you have more enzymes, the reaction is faster. So by reducing the amount of enzymes, we can slow down the speed of reaction. But there's one major drawback with this type of control mechanism, which is it is very slow. Imagine if you need more enzyme, your cell will first have to transcribe the gene into more mRNA. Then it will be translated in ribosomes and be modified in Golgi apparatus to become a final functional protein. So this process might take a few minutes, which is considered slow for molecular processes. While a second strategy, which is faster, is by controlling the activity of an enzyme through allosteric binding or allosteric control, which can happen in milliseconds. So in this first type of allosteric control, you have the binding of effector molecule, which could be ATP or any other metabolites molecule that can modify the structure of the enzyme. So sometimes the binding of the effector molecule can switch on the enzyme, or sometimes the binding of the effector molecule can switch off the enzyme. So it all depends on the type of mechanism and reaction we are talking about. While another type of enzyme activity control is known as covalent modification, such as phosphorylation. This type of control will require the help of other enzymes such as kinase to add a phosphate group to the enzyme or phosphatase to remove a phosphate group from the enzyme. Again, sometimes the addition of the phosphate group can switch the enzyme on, or sometimes the addition of the phosphate group can switch an enzyme off. It all depends on the type of reaction we are talking about. Another type of control is by controlling the interaction in between enzyme and the substrate, such as compartmentalization of enzymes in an organelle, such as mitochondria. So for example, by concentrating all enzymes within the mitochondria, we can have a better efficiency in the citric acid cycle. On the other hand, we can also compartmentalize the substrate, such as glucose, inside a cell with the use of a hormone, such as insulin in this case. So such control mechanism is known as hormonal control of metabolism. Okay, so far we have talked about some general strategy of control in metabolism. And now let's look at some real examples of control in glycolysis. So showing here is the glycolytic pathway, and there are three important enzymes involved in the control of glycolysis, which are hexokinase, PFK, and pyruvate kinase. So PFK or phosphofructokinase is the most important enzyme in the control of glycolysis because it is the rate limiting step. So what does it mean to be a rate limiting step is that PFK has the slowest rate of reaction among all these enzymes. So when it is the slowest enzyme, it will determine the pace of the whole glycolytic pathway. And PFK is activated by the presence of AMP or the adenosine monophosphate molecule. AMP is formed when our bodies have a low energy level. So if you look at this reaction, two ADP can be joined together to form an ATP for energy supply, and it comes with a side product which is an AMP. So whenever we have increased AMP level in the cell, it signifies we need more energy, and hence the PFK is activated by AMP through conformational changes to stimulate the following pathway. And another activator for PFK is fructose 2,6-bisphosphate, which is produced from fructose 6-phosphate. So when you have the presence of this fructose 2,6-bisphosphate and AMP, it activates the PFK, and you will get more of the ATP production. What a second enzyme in this glycolytic pathway control is pyruvate kinase, and it is stimulated by the binding of fructose 1,6-bisphosphate. And this process is known as a feed-forward stimulation, as the feeding of the substrate actually modify the enzyme activity. But what will happen when we have high energy levels in the cell? Well, the glycolytic pathway will be inhibited. PFK, for example, is inhibited by ATP, which is a high energy molecule. So it is also inhibited by citrate molecule, which is a substrate from the citric acid cycle. So I'll explain later why citrate can also inhibit the glycolytic pathway. While another factor inhibiting PFK is a low pH. Now, if you look at the final product of glycolysis, you have pyruvate, which is better known as pyruvic acid. So when you have a lot of pyruvate or pyruvic acid, the pH is going to be reduced, and this signifies that the cell have enough energy, and hence PFK is inhibited by a low pH. And for pyruvate kinase, it is inhibited again by the ATP, while for hexokinase, it is inhibited by G6P in the process known as a negative feedback. So the whole idea of enzyme inhibition is to stop or to reduce the rate of glycolysis to prevent energy wastage.